First of all, I'd like to thank the organizers for the invitation and the opportunity to tell you about the pantazines. These are a series of small drug-like molecules that stimulate CoA synthesis with a promising potential for PKAN therapy. CoA is vital for many essential functions. CoA is a carrier. It provides molecules to generate energy and maintain a balanced environment to support function. CoA derivatives also supply the precursors for cellular building blocks that are necessary for growth. And we learned from Dr. Sibon earlier in this session that CoA casts an even broader net by donating activating prosthetic groups to proteins. Panthenate kinase, called PAN-K, controls the rate of CoA biosynthesis. PAN-K controls the first step in a 5-step process leading to CoA. How is the activity of PAN-K regulated? When CoA derivatives or thioesters build up in the cell, these bind directly to the PAN-K protein and inhibit activity. Now, acetyl-CoA is the most abundant CoA thioester in the cell, and that's why I've shown it here. And under normal circumstances, about 95% of the PAN-K protein is bound to acetyl-CoA and turned off. All PAN-K proteins have the same overall configuration, with 2 complete peptides interacting to form a dimer along the length of helical structures running down the middle of this diagram here. Now, the lower panel shows an open configuration, and this is the structure of the PAN-K3 protein in the inactive state. You can see acetyl-CoA depicted with a ball and stick structure stretching across the dimer interface, holding the protein open. Now, there are 4 active PAN-K proteins encoded by 3 separate genes. All 4 PAN-K proteins contribute to CoA synthesis and are regulated by CoA, but with different sensitivities. At least 2 to 3 PAN-K genes are expressed in cells, and PAN-K3 is special because it is ubiquitously expressed. That means it is constant and expressed in all cells and tissues that we've looked at. And that's one reason why it was chosen as the primary target for our drug development. We generated several mouse models of CoA deficiency, and our most recent models, down toward the bottom of the list there, had movement disorders. CoA is variable and responds to different metabolic challenges by changing its activity and changing the level in cells. So it's hard to really know what is a CoA deficiency. Along the way, we learned that both PAN-K1 and PAN-K2 contribute equally to maintaining brain CoA levels in mice. And in the inset panel up on the upper right, you can see the transcript levels for both PAN-K1 and PAN-K2 in mouse brain. Now, that we realized, seeing that it was necessary to reduce both PAN-Ks to measure a reduction in brain CoA using our biochemical techniques. And these early models were too severe to be useful. And I'm not going to give you those details. So we took a step back and went from whole body deletion to deletion of one and then both PAN-Ks, specifically in neurons. As the CoA impairment became more localized to neurons, the mouse models lived longer and showed a movement disorder. And I will be talking about the last 2 models in this presentation. We obtained new insight into the molecular pathology related to a CoA deficit in neurons in the model That is the whole body deletion of PANK1, and PANK2 was deleted in neurons only. In wild-type cells, in wild-type mice, I should say, we learned that CoA actually increases fourfold during brain development, during the 3 weeks after birth. However, in this model, the brain CoA does not increase, and the neurons become deficient. The CoA deficit impacts free CoA, or CoA-SH, that's listed here. That it means it's an unbound carrier, and it's free to take up and ligate with the necessary molecules around the cell. It also impacts the short-chain CoA thioesters, such as acetyl-CoA and succinyl-CoA, shown here. And these are necessary for energy and ATP production. Long-chain CoA thioesters were not affected, suggesting that lipid synthesis and metabolism was not a proximal target of the CoA deficit. A full-scale analysis of gene expression in brain using RNA-seq revealed that expression of the genes in heme biosynthesis were reduced in the model. And that includes the aminolevulinic acid synthase and the brain hemoglobins 1 and 2. Now, succinyl-CoA is the precursor for heme synthesis, and so we measured heme directly. We found a 50% reduction in heme in brains. And this is quite significant because heme is an important cofactor in iron metabolism and also activates selected proteins, including fatty acid hydroxylase, which is a heme-binding protein. And interestingly, mutations in fatty acid hydroxylase lead to another NBIA disorder called FAN. Now, when the brain CoA deficit occurs during development in this model, a problem with movement becomes evident. Here we show a pattern of movement of mice during a 5-minute activity period in a closed arena. The activity was monitored with video, and we can see irregularities in the pattern of the movement of the model compared to the wild-type matched animal. This movement included pauses with intermittent shaking, and the shaking was recorded as movement. So there's no apparent decrease in movement, but there's a substantial decrease in the distance traveled by the animal over a 5-minute period. We also saw consistently forepaw crippling, which was reminiscent of what we see with PKAN patients. And this was also associated with an absence of strength in the forepaw grip. So what are pantazines? These are drug-like small molecules that bind directly to PANK protein with high affinity. They counteract the feedback regulation by the CoA thioesters and activate PANK and thereby stimulate CoA biosynthesis. The pantazines lock the PANKs in an active conformation, making them refractory to feedback inhibition. And this is a unique mechanism of action, unique among any drug that we know of. The pantazines are orally bioavailable and readily cross the blood-brain barrier. Now, pantazine development started with a high-throughput screen many years ago at St. Jude using our in-house chemical library. Purified PANK3 protein was used as the protein target, and we measured PANK3 activity in the presence of each of more than 500,000 compounds. Out of this, there were 1,000 compounds that were identified as identified as potential modulators of the activity. We knew this was possible because through the years, we've actually found naturally occurring activators of pantothenate kinases, namely long-chain acyl carnitines and acyl ethanolamides. Now, these 1,000 compounds were chosen. They had mid-range potency. They weren't the best hits in the screen, but they were mid-range because we wanted something that we could improve to work with. We computationally screened these compounds using two parameters. We wanted a low molecular weight, so we had plenty of room to modify the molecule chemically, and we also wanted good lipophilic efficiency, which enhances blood-brain barrier permeability. And these parameters are among the characteristics of the top 25 central nervous system drugs which cross the blood-brain barrier. And the reference at the bottom of this slide was where this principle came home to us. This computational screening narrowed down the candidates to 100 compounds, which interestingly segregated into 8 unique chemical scaffolds. And we chose one, called it pantazine 2789, and decided to work with it. Starting with that initial scaffold, we synthesized about 200 compounds and introduced different chemical modifications that were guided by our detailed knowledge of the pantazine binding pocket in the PANK3 protein structure. The right panel, you can visualize a PANK3 protein saturated with pantazine 2891 in pink, binding across the interface between the protein monomers. Each new compound after 2789 went through a battery of tests, a lot of different chemical techniques at the bench, coupled with testing in cultured cells and animals. And in the course of this process, we increased the potency more than 400-fold. We extended the lifetime of the pantazine in circulation 3-fold. The absorption in the GI tract reached more than 95%, and there was a bit of formulation development involved in this aspect. All the while we maintained an ability to cross the blood-brain barrier. And interestingly, several of our modified compounds lost this ability by just changing one atom in the pantazine structure. Each improvement allowed us to lower the dose that would be effective in elevating brain CoA, reduce the dosing schedule, and establish a wide therapeutic index to establish safety. To illustrate how pantazines work, we treated wild-type mice under normal conditions with high-dose pantothenate, shown in the panel on the left. And this alone did not change the brain CoA levels. Pantothenate was delivered by oral gavage, and brain CoA was measured 4 hours after dosing. However, co-administration of pantazine with the pantothenate, shown in the right panel, demonstrates how the pantazine opens the gate for CoA biosynthesis, and elevation of brain CoA corresponds to the pantothenate dose. And here we've saturated it. Now, my slide. Yeah. Moving to our latest mouse model with deletion of both PANK1 and PANK2, specifically in neurons only, we see that the animals have an abbreviated lifespan and remain small after weaning. The panel on the left shows the lifespan in black, and on the right, we see the weight gain with time after birth on the right, in the right panel. Now, we treated these animals with the pantazine 2891 delivered in the chow. We increased the lifespan of the mice 3 times and enhanced the growth of the mice as we measure the gain in body weight that is shown in red in the right panel. Movement in this mouse model also improved following pantazine treatment. If you look at the video tracing of mice in the open field test, on the left panel, we have matched wild-type mice moving freely in the arena over 5 minutes. And treatment with pantazine did not alter this behavior. In the right panel, the mouse model with 1 and 2 knocked out in neurons only, the mouse model had serious issues with movement and had intermittent tremors. Treatment with the pantazine significantly improved the locomotion of the animals. And this behavioral test was done just before we harvested the animals to measure brain CoA levels. The brain CoA levels in this model were significantly reduced compared to matched wild-type control animals. And this is a total CoA measurement and includes free CoA as well as CoA derivatives. The pantazine treatment returned the brain CoA to normal levels. And as I mentioned before, these measurements were performed at 45 days after birth following three weeks of treatment. So I'd like to summarize and point out a few things. PAN-Ks are the control points in CoA biosynthesis, and CoA levels adapt to metabolic challenge by modulating these PAN-K activities. Mice have both developmental and functional dependence on CoA. Now, pantazines bind to all PAN-Ks, not just PAN-K3, but with different affinities, and I haven't had time to show you that data. The pantazines can stimulate CoA levels in multiple tissues, and pantazines have excellent oral bioavailability and efficiently cross the blood-brain barrier. We've partnered with a company, CoA Therapeutics, which is a subsidiary of Bridge Biopharma. CoA will be moving a pantazine forward to clinical trials. The therapy is active in the brain and designed for convenient oral administration. The US FDA granted CoA Therapeutics rare disease, pediatric disease designation for PKAN just earlier this month. And CoA Therapeutics plan to initiate the phase 1 studies in healthy volunteers for this first-in-class drug in 2021. And plans are, we anticipate having the clinical trials in PKAN patients in 2022. Now, this was a team effort, and I was fortunate to have colleagues who specialize in key areas for hit-to-lead progression and in structure-guided design for drug development. I handled the biological efficacy. And we would also like to acknowledge financial support for this project through the years, in particular, LSAC at St. Jude, and most recently, CoA Therapeutics. And I'll stop there, and I'd be happy to answer any questions. So I guess I'm moderator, so I have to look at the questions. Do I have questions?
Susie, there is one question in the chat, actually, not in the Q&A, but in the chat. I can...
Let's see.
It's a question by Fatime Mollet.
Oh, yes.
How long were the mice treated with pantazine to improve their movements as you showed?
Those mice were treated for 3 weeks. And that was just the time point that we chose to look at.